Hi guys, it's Simon C. Welcome to this new AI Radar. Today I have for you 3 more artificial intelligence news about AI in healthcare. In a new blog post, Google researchers wrote that they recently presented Parrotron, an ongoing research project that continues and extends their effort to build speech technologies to help those with impaired or atypical speech to be understood by both people and devices. Parrotron consists of a single deep neural network, say the researchers, trained to convert speech from a speaker with atypical speech patterns indeed directly into fluent synthesized speech without an intermediate step of generating text. Parrotron uses only speech signals and doesn't need visual cues such as lip movement. Specifically, the first training phase uses a corpus of 30,000 hours that consists of millions of anonymized utterance pairs. The second training phase utilizes a corpus of utterance pairs used to adapt the language patterns specific to the input speaker. Google researchers claim that Parrotron can help people with a variety of atypical speech patterns, including those with ALS, deafness, and muscular dystrophy, to be better understood in both human-to-human interactions and by speech interfaces, with its end-to-end speech conversion approach more likely to reproduce the user's intended speech. Researchers discuss further application of Parrotron in their paper published on archive.org and provide audio samples in the dedicated GitHub repository. Megan Scudellari reports on Spectrum that surgeons and computer scientists at Johns Hopkins University have built a test called CompCyst for cyst analysis that is significantly better than doctor observations and medical imaging are predicting whether patients should be sent home, monitored, or undergo surgery after being diagnosed with pancreatic cysts. Actually, up to 78% of cysts for which a patient referred to surgery end up being not cancerous. So, to identify patterns to distinguish low-risk from high-risk cysts, the authors trained a machine learning algorithm called MoCA for multivariate organization of combinatorial alterations, which combine molecular data with protein information from extracted cyst fluid and imaging tests of 436 patients diagnosed with a cyst, who then underwent surgery to have it removed. The team then tested the algo on a separate set of data from 426 patients. What they found is that CompCyst outperformed the standard of care that doctors use today in all 3 patient groups. Indeed, the test correctly predicted 60% of patients who should have been sent home versus 19% using standard of care, 49% of patients who should have been monitored versus 34%, and 91% of patients in need of surgery versus 89%. Overall, the researchers estimate that if CompCyst had been used to decide care for these patients, 60 to 74% of them, depending on the type of cyst, would have avoided unnecessary surgery. The study was described in the journal Science Translational Medicine. Additionally, senior author Anne-Marie Lennon expects to offer the test within 6 to 12 months at the Johns Hopkins Kimmel Cancer Center and hopefully to make it commercially available after a larger prospective clinical trial. Combining clinical and genetic features using machine learning is the wave of the future to inform clinical judgment, not only about pancreatic cysts, but about many other diseases, said co-author Bert Wolgerstein. On Business Insider Australia, Sharon Mazig reported that researchers at Australia's Flinders University developed a new vaccine believed to be the first in the world to be designed by artificial intelligence. An algorithm called SAM, for Smart Algorithms for Medical Discovery, being named so after the goal of searching the universe for all conceivable compounds to find a good human drug, also called a ligand. Flinders University professor Nikolai Petrovsky led the team to train the artificial intelligence program on a set of compounds that activate the human immune system and a set of compounds that don't work, to discover on its own the patterns that separate a drug that works from one that don't. Then they developed a synthetic chemist, a program which generated and fed to SAM trillions of different chemical compounds so that the algorithm could find good candidates for being suitable human immune drugs. The team then synthesized those candidates in a lab and tested them on human blood cells to see if they would work in boosting influenza vaccine effectiveness. This confirmed that SAM not only had the ability to identify good drugs, but in fact had come up with better human immune drugs than currently exist, Petrovsky said. He also said that this approach potentially shortens the normal drug discovery and development process by decades and saves hundreds of millions of dollars. Okay guys, it's all for today. See you next time with AI Radar. Bye!